Hi, this is Jennifer Jones. 727. We're talking about the direct and negative stains. So this week we had, let me show you I gathered all my supplies needed for the material of this lab, microscope. And then I have lab 1 and lab 2. So in lab 1, we actually had to label the slide direct 1M and circle on the opposite side so that when we apply the cheek and the gum area onto the slide, then we know where the area is actually located. We also used the candle for the heat so that we could kill the bacteria so it did not spread. Let's see. Then I placed the slide that I was looking for into the staining cup, which was like half of a bottle. I threw that away already, sorry. So then we placed 2 drops of the crystal violet, which is going to be this one, stage 1 crystal violet. And you kind of see it turns a little bit purplish on the actual slide. So once that sat for 16 seconds, then we rinsed it off with water and allowed it to dry. Photographs were taken. Then we went ahead and got a different slide, labeling it number 2 and negative. I guess I didn't put a circle, but. And for this one, we actually used a couple drops of the iodine. That would be just number 2. Once that happened, I would then use this extra slide to kind of like shimmy it down, spread it across the initial slide so that we could have better view and distribute it properly over the slide. Came out like a little bit of a reddish tint, like kind of like dried blood. And then we looked under the microscope and saw all of our samples from my mouth. Yum, that's kind of gross. This one is not here. So yeah, this week is this was lab number 1. And then we have lab number 2 over here, where I actually did it about 3 times. One for the E. coli, one for the S. cerevisiae, and then the other one for the S. epididymis. In this process, we actually used 1 to 2 drops of the crystal violet, allowed it to sit there for 60 seconds, washed it off with water. Then I put 2 to 3 drops of the iodine, let it sit there for 60 seconds, washed it off with water. Then we did the actual decolorization, where I actually saw everything go clear, but then it really came back. Let's see. After that one sat for 30 seconds, and then we went ahead and did the saffron for 30 seconds as well, rinsed, and then we have these were all examined also underneath the microscope. Good thing I still had my original samples from lab 1. These were all heat treated using my handy dandy close clip, and I would hold them. Let's see, I can't even do it with phone. I hate phones. Hold it as so, and then just pass it over about 5 times. Not too much to burn the glass and not too much to make it too hot, just enough, and then allowed it to cool. And then again, yeah, we looked at into the microscope for our results.